Hello to everybody. We will start the fifth session of the Congress with a keynote speech. I invite Professor Ahmed Uludak to do the presentation.
Good afternoon. Actually, it's about the Uppsala region of Edirne. Edirne's town, district of Uppsala. But probably I will talk more about the general rice wheat problem in rice in Turkey. Why we choose the Uppsala region? It's one of the biggest rice growing area. Most of the fields are rice fields there. Roughly, that's just a town, and maybe 6,000, 8,000 hectare area is allocated for the rice production. I have two co-authors, and Ali Galip is my undergrad student. He is graduated this year. Deniz İnci worked with me in his master's in Turkey. Now he's doing his PhD in California, Davis, and he also works on the rice in California. Also, he's here, and he will talk after me, probably. So this is how the world rice areas and rice production. And if we look at this from 1961 to today almost, and rice production area is increasing, and production also increasing. The blue line is the rice production area in the world. I'm not good at it to remember the numbers, but so it is getting larger and larger, but I don't know, after that we will be able to increase it. So this is the maybe more showy picture, and darker the color means higher the production. So if we look, it doesn't show here probably. Because, no need. Turkey is not one of the highest producers of the world, but the rice is very important in Turkish farmers, especially the farmers in, okay, let's first talk about this, then later I'm gonna talk about the areas specifically. So this is the same graph. The first one was about what's going on in the world, and this is what happens in Turkey from 61 to today. And long time, almost until 2000s, rice areas never expanded. So after 2000, demand in Turkey increased, highly increased. Then depending on the demand, also rice production areas increased in Turkey. Still, we import huge amount of rice. It used to be before from United States, but now we are importing the rice from other countries. So we were importing from United States. California Calrose was very cheap that time, but now Japan pays for the Calrose huge amount of money because it's a very good rice variety for the sushi. So we couldn't pay that much for the rice, unfortunately. There is no number here, but when I compare the yield in Turkey with the world average, we are over the world average. And many farmers in that area, especially in Edirne area, get the 1 ton per hectare, no, 10 tons per hectare. 10 tons per hectare. So, and most farmers gets the over 6 tons from per hectare in Turkey. So the average is really high. Let's look at where we grow the rice. And I don't come here. So this area, we are here in Edirne. And this area, almost including all this region, 70% of rice production of Turkey comes from that area. And maybe 30% mostly comes from that area, usually our region. And the other ones, generally very small areas and local varieties, some of them custom local varieties. But yeah. This Edirne area, Marmara region of Turkey, I will tell it again, maybe someone was not able to hear me, and 70% of the rice production in the top near Edirne area, all this area. Edirne itself almost produces the half of the production in Turkey. And Edirne is the top province. So and then the other darker color area, it produces more or less the 20–25% of the rice, and the remaining only 5%, around 5%. But they have some local varieties, and the people, especially local people, prefer that varieties because the taste is different a little bit than comparing to other rice. Similar map. This is the production map. The first one, the area map, and this is the production map. No, excuse me, both of them the production map. And as you see here, this is just to show the better how big or how much Edirne produces the rice, and followed by the Samsun. But this area still produces less than Edirne. So, I do not have time, so maybe you can remind me. I will talk half an hour. Half an hour for me allocated time, or 15 minutes? Half an hour, okay, thank you. Actually, in Turkey, the biggest problem, as in many countries in the field, weeds. We have the problem of water. We have some other things. But the biggest problem is the weeds in the field in Turkey. And today, we have the water grass problem, echinoclea, and in Turkey, in rice fields, it's reported 5 different species is seen in rice fields. Probably we have more species, but not common in rice fields. And these species, and my students worked the last 3 years in the rice fields. They visited several fields and different parts of the area, this Marmara region. And, oh, I should show where I live, actually. Okay. Here is the Çanakkale. I work here, Çanakkale. Also, we have the rice production. And my students visit the fields in Balıkesir and Fatih, Bursa, Çanakkale, and Edirne. And they check which echinoclea we have. And in that area, in our area, we have mainly Echinoclea oryzoides. And the second large population is the Echinoclea orzicola. But today, this people dealing with this, botanists dealing with the name of the plants, they combine the orzicola and oryzoides as a one species and called the oryzoides. But farmers differ it. We differ it. Why? The reason is orzicola comes later. If you go to rice field today, we will see oryzoides all mature seeds, and they mostly fall down. And if we look at the orzicola, they are still green. They are not matured yet. Almost 2 weeks difference between these two species. I mean, we follow the farmers because they have the problem with this late water grass. And so the name is the late water grass. We have Echinoclea cruscalli also. And before, we always thought mostly we have the Echinoclea cruscalli, but not in rice fields. Rice fields, we have oryzoides. Maybe this is the reason the Echinoclea cruscalli cannot grow under the water much. So the rice in that area mainly classical way, and then we keep it almost all growing season under the water. Sometimes before applying pesticides, we make it dry, or in the beginning we make it dry to let the rice catch the soil with the roots. But it's always under the water. Nowadays, due to the shortage of water, and drip irrigated rice is supported by the government. And some areas, farmers produce other crops. They put the rice in their rotation system now. But it is a very new system, and in the beginning, first 1 or 2 years, there is no much weed problem, but after 3rd year, it starts immediately. And not the same weeds, but Echinoclea cruscalli is definitely there, because it is good for the Echinoclea cruscalli. And some other weeds seen, we will not mention today, like Xanthium species, they are not seen in the rice fields, but we see them in the summer crops, cotton, maize, whatever, also in vegetables. So we saw it. But now, problem will change if we increase the drip irrigation. But we doubt about it because most of the rice production area is kind of flooded area. It originally gets the floods. So then it is much more easier, and water table is very high. So it's much more easier to keep the current species. So Echinoclea species, I've told about this, and I told you the 5 species all over Turkey, but in this area we found the, I will repeat it, Echinoclea oryzoides mainly, and then Echinoclea oryzicola, Echinoclea cruscalli. Few fields, maybe 1 or 2 fields, we found the Echinoclea colonna. And we found some species we were not able to identify them correctly. We thought they are also the Echinoclea cruscalli, but maybe needs more study on it, and we will check the chromosome numbers because they have different chromosome numbers, so we will check the chromosome numbers of our samples. So another important species, Leptohlea fusca. Leptohlea is increasing, and we can see it in many fields, but it's still increasing and getting more and more important. And Sparus species, especially Sparus deformis, is a very common species in that area. Also, alisma and some other broadleaf species are common. That's when you ask the farmers, generally they will tell you they have a Echinoclea problem, and now we have another problem increasing, VD rice. VD rice is totally different from the wild rice. It's a kind of spin-off normal rice, and it comes from rice we eat. What we did, we followed the 3 fields, and we got the data from these fields, especially the farmers helped us to get the data. Why we did this study? Because we would like to show how detrimental will be the weeds in rice in Turkey. Because for farmers, if they have an herbicide to control something, so they don't care. Okay, there is an herbicide and it's not very expensive, it is good and you earn when you apply the herbicide, so they just apply herbicide and they do not think much. Now, in this situation, farmers also started to think because herbicide choices is getting less and less. Some of them is prohibited, and in Turkey or in European Union. So, and some of them getting the resistance, and weeds are getting resistance to some herbicides. So the problem is getting bigger. So when we look at here, the first applications, this is the 3 fields, you see the fields, and we only put here this presentation, herbicide applications. We also asked the fertilizers and some other things from these fields, even we have some data, yield data, but we just wanted to make it the problem part of this study. So the first line is the pre-application. Just we applied herbicide before rice. Ektikten sonra uyguluyoruz. Yeah, I made a wrong sentence. We seed the rice, then we apply it. And the other two is the post applications. When you look at this 2018 data, first field gets the second application of post twice, 2 herbicides mixed together, and another field gets the 3 herbicides twice. So see, and wheat problem is increasing, and then this field is the field 2 is the using more herbicides. And if you look at the field 2, they have the 4 herbicides mixed, tank mix. It's a huge amount of herbicide application. But farmers need it. Otherwise, they cannot, I will say combat. Normally, I do not use the combat word for the weed science, but the combat with weeds. And 1 year later, more or less the same scenario, but this is a little bit depend on what the farmers see in the field. Like the field 2, it was more weedy than the 2019. So the farmers did not apply these 3-4 herbicides together. But if the problem gets bigger, they will apply more herbicides. Like the same field 2, applied the huge amount of herbicides together. It's kind of, we do not know what's happening there. We get this synergistic effect, or we get some other things. We do not know. And what's going on in the nature? Because it goes to the water. But you cannot measure what's going on in the water because they will be different. So probably this field that year got a huge amount of problem. And the following year, same scenario in the same field, and the field 1 also have the biggest problem. And for example, some of these herbicides, we have the rates also here. Let me go to the first one. And when we look at the rates of the herbicides, it's increasing actually in many cases. And so farmers use more and more herbicide and mix of herbicides, and we have the 2022 data. And as you see here, also we have huge mixes and a huge amount of herbicide application, always increasing rates. And as you see, pre-applications also become 2 herbicide. Now, I would like to conclude something. Pardon me, I forgot to tell this. As you see here, 3 times post-application. So what's changing? We mix more and more herbicides. We increase the rates. Also, we increase the time to apply the herbicide. actually, farmers maybe will apply more times, but there is no time for the growing season ends. Otherwise, they would like to apply it. Because if we go to the fields now, we will see the many echinoclea species on the fields, and even they get this much herbicide application. And not only echinoclea, but also other species can be seen. But the biggest problem in our area is the echinoclea main problem. Now, as I told the beginning, farmer is happy with the herbicide application. Even they apply many times, but they pay and they get. They pay for herbicide, they get money from the rice, because without herbicide, they cannot get any crop probably. It's sometimes, if they leave it as it is, and they will not get the maybe 100, they will get maybe 100 kilo, 1 tons per hectare. We use the decar, and generally I remember the decar, so the hectare is the 10 times more than decar. Farmers, if they get 80% of control, they are happy with these herbicides. When we registered herbicides in Turkey, and registration committee looks for the effect 90%. So if an herbicide gives the less than 90% effect, so it cannot be registered in Turkey. So, but the farmers happy with the 80%, but it goes down to 80%, so it's a huge problem for farmers, because huge amount of weed we have in rice fields. Some people think drip irrigated rice will solve the problem. No, it will not. And not only weed problem, because drip irrigated rice gives the less yield, less crop. So farmer can get the almost 10 tons per hectare. So 10 tons per hectare getting farmer from this flooded rice, traditional rice, and then we'll get the same field, maybe 600 kilo, same time, the same year. So they will not do it. If there is a water, they will continue this flooded rice system. And they cannot change the crop, they cannot rotate the crop in these rice fields, most of them. Because most of them, as I mentioned, with the higher water table, and they are normally flooded areas, and they are like Edirne, and so they call it the 3 rivers meet in Edirne. So these rivers have the delta, and all these delta areas are rice mostly, except a few of them. So all the area, if there is a water, they will do it the flooded method. So we need to find something else, because we haven't checked the herbicide resistance in our area recently, what's going on, because farmers are increasing the rates, but after a certain point, they cannot do that. So maybe they do not care, they know they have the herbicide resistance. They know it, because they are increasing the rate, and they know this is the herbicide resistance weed in their field, but still the rates applied bearable. If they can apply the bearable rates, so they do not care. But at the end, we will reach the sea, so there is nowhere to go. And I think I talked much today, and I would like to finish here if there is a question. Thank you for listening.
There are some questions for Professor, please.
Thank you for talk. I wonder.
Closer.
Okay. I wonder, quincularak, the herbicide. You know, the quincularak is forbidden in Europe, but in Turkey is quincularak legibility is high and drain flow not good and moderate toxic for aquatic organism. What do you think about this herbicide?
Actually, quincularak was prohibited in Turkey before, and they get the temporary permission for quincularak because they couldn't solve the problem, weed problem. Now, quincularak helping, but not more. It will be bigger problem. It will be bigger problem, and we prohibited it before. Then farmers pushed the government. They need a solution, and they know the quincularak is good for them. So I think it's the temporary registration has now, but I don't know how many years they can keep it. Even the farmers will not enjoy it at the end. And in my area, tomato is this most important crop in the Çanakkale. And farmers get the quincularak problem in tomato. And last year, one of our students worked on how we can heal the tomato affected by quincularak. So we did this also master thesis, yeah. I agree.
Other question? No.
Thank you.
Thank you, Professor. Of Ambriesus Surski as biological control agents. I invite Seren Dogan to do the presentation.
Good afternoon, dear Chairman and participants. I am Ceren Doğan from University of Uludağ, Turkey. I present my master science thesis about artificial developing pesticide resistance of a biological agent. We start. Why did we decide this study? There are two important reasons. Firstly, the economic value of pepper production for Turkey is quite high. The other main reason is unsuccessful control for insects and mite pest of pepper. Since the common method for pest control is spraying with some synthetic pesticides, the method causes destruction of non-target organisms, chemical residue problem on pepper products, resistant pest to pesticides. The two-spotted spider mite is one of the main pests of pepper. I'm sorry. This species is the most resistant species to pesticides among other arthropod pests. Using intensively acaricides for control of this pest leads to pesticide residue problem. The answer to the question, what is ecological solution in mite control, is biological control for sustainable production. Amsur sibirski is considered as the most effective biological agent of spider mites. Commercially, the predatory mites commonly sold in Europe and North America. Also, Amsur sibirski is controlled the other main pests such as thrips and whiteflies. But the registered acaricides in Turkey are very toxic to commercial strains of Amsur sibirski. Sometimes we have to use acaricides for a reduced high spider mite population. Unfortunately, these applications cause destructive to Amsur sibirski population. As a new approach, can we use pesticide-resistant Amsur sibirski population? As you can see this table, there are some evidence about pesticide-resistant of phytocyte mice in previous study. Interestingly, there is no record about pesticide-resistant to Amsur sibirski population. The aim of this study were to test side effect of bifenthate and abamectin mixture for a nature Amsur sibirski population, to gain resistant bifenthate and abamectin mixture with artificial selection under laboratory conditions, to detect side effect level of these acaricides against resistant population after 5 selection. Material methods. This study carried out in our archaeology laboratory and climate chamber. In a controlling climate room, we grew up bean and pepper plants from seeds as sterile. A Turkish strain of Amsur sibirski was much reared with Carpophilus lactis and Tifilatifolia pollens within petri dishes in a climate room. We have to use 2 acaricides, modification acaricides. Abamectin is nerve poison effective acaricides. Bifenthate inhibitor energy metabolism effective acaricides. Firstly, the pepper we collect the pepper plants. Then applied determined acaricide concentration on buildings with a spring tower. The leaves were put in the Minger cells. Then added 100 spider mites eggs on buildings leaves. Some pollen and close to Minger cover. The Minger cells were put in a climatic chamber. The mite vitality was checked out after 4 days. After counted new laying Amsur sibirski eggs, at the end of experience, the monitored vitality of meat emerged during its until matured. At the end of experience, the lethal dose of acaricides for adult and juvenile for Amisor risk calculate by using probit analysis. Mortalities of adult and juvenile and reproduction reduction rates of Amisor males was determined by using for the recommended 5 dose compared with control individuals. The combined total adverse effect of the acaricides of Amisor risk was calculated using the formula. The A results were rated into 4 classes like this. Results. Vertical axis shows DTRH percent and horizontal axis shows Amisor risk adult population as 1, 2, 3, 4, and 5 after 5 selections. After the first selection, the DTRH of Amisor risk significantly increased. Among selected population, the fifth population significantly more resistant to the recommended 5 dose. Similarly, juveniles of fifth population significantly more resistant to the recommended 5 dose. You can see egg-laying results in this graph. Egg-laying Amisor population significantly increased after the first selection with the acaricides. In H5 population, significantly higher numbers of egg-laying per female were observed compared with the native S1 and S2 populations for the recommended 5 dose. You can see preconception rates of Amisor risk adults. The rate significantly more increased after the first selection with acaricides. More pre-feeding behaviors were observed in selected adult population compared with native population for the recommended 5 dose. Similar significant higher preconception rates were determined in Amisor juveniles of the selected population after the first selection with the acaricides. You can see lethal concentration of Amisor adult graph. Lethal concentration of the fifth adult increased compared with those of native adult population. After 5 selection, the adult might gain resistance approximately 20-fold. Similar higher lethal concentration the last, Similar higher lethal concentration the last 2 juvenile population were observed compared with native population. After 5 selection, the juvenile mice gained resistance approximately 55 folds. You can see side effect change of risky adults after selection in this graph. While the recommended dose was moderately harmful, for adults of native population, the dose even more were found harmless as 3, 4, and 5 populations. Similarly, the recommended dose is and consecutive higher doses were harmless after the 5 selection. As you can see this figure, the adults might develop resistance to the recommended 5 dose and upper dose of the acaricide mixture. Similar results were observed in juveniles. Ongoing studies. Nowadays, we are examining fitness parameters of the resident Amisor risk population compared to those of the native Amisor risk population. We focus on reproduction and development performance of the resident Amisor risk population. The results show that Amisor risk can easily develop resistance to the commercial pesticide of abamectin and bifenazate mixture. After 5 artificial selection with acaricides, the resident population can consume more prey and lay compared with the native population. For sustainable control of spider mites, the resident biocontrol agent may be used together with acaricides in the future, but the results must be approved in field conditions. Thank you for your attention, and thanks to Dr. Agribalkan.
Thank you, too. There are questions for Seren? No? No, no questions, so thank you again. The next presentation will be determination of some morphological characteristics of herbicide-resistant M3 mutant quinoa, Chenopodium quinoa wild lines. I invite Omer Egritas to do the presentation.
Hello everybody, I am Ömer Ayırtaş. I will present my study to you about determination of some morphological characteristic of herbicide-resistant M3 mutant quinoa lines. It have 3 parts. One part about generation information quinoa, two part material and methods, 3-part conclusion. Ancient grain quinoa. Quinoa is an important crop, is resistant to salinity, can adapt to harsh climatic conditions. Is gluten-free and has seeds rich in nutrition. It's estimated that agriculture has been practiced by Central and South America, American Indians, since 3000 BC. NASA uses this plant in the diet of astronauts. Situation in the world and in Turkey. There is 200,000 hectare of cultivation area, almost all of which are in South American countries, and total of 250,000 ton grain production per year. Intensive studies are being carried in the USA, China, India, the Netherlands, the UK, Germany, Denmark, Finland, Serbia, Greece, Lithuania, and Pakistan. Approximately 12,000 hectares are cultivated in Turkey. Use of quinoa. The main use of quinoa is in human nutrition. It is also used in animal nutrition and in industry. There are many ways of use in human nutrition. It's possible to use all kinds of bulgur and rice. Why is it important? Its popularity is in recent years is due to it is important in human nutrition. Quinoa seeds are high nutrition and suitable for the taste of the people of our country. Quinoa seeds are rich in protein and amino acid. It's a storage of minerals. It's good source vitamins A, B, C, and E. Quinoa is a plant high altitude. It grows from sea level to 4,200 meters altitude in South America. Since its water consumption is low, it can be easily grow in arid conditions. It is a plant of salty soils. It can survive in seawater salinity. The most important problem is quinoa farming. The most difficult aspect of quinoa cultivation is weed control. Since there is no selective herbicide for quinoa cultivation, It is necessary to develop herbicide-resistant quinoa lines. Aim of the study. This study was carried out to determine some morphological characteristics of herbicide-resistant mutant M3 generation quinoa lines under greenhouse conditions. Material and methods. In the study, 4 mutant lines and control plants were planted in pots according to completed randomized experimental design. When the plants maturate, some morphological characteristics such as plant height, stem thickness, and number of branches were determined. Mutant quinoa's genotypes. Titicaca variety of quinoa was used in this study. In order to develop quinoa lines resistant to imidazolone herbicide, the method used in development of herbicide-resistant wheat in the USA was used. Seeds were treated with sodium azide as a chemical mutagant, and these seeds were sown in field and sprayed. Seeds were taken from the surviving plants and sown again. Plants in this generation were all sprayed, and M3 generation seeds were taken from surviving plants. Results. According to these results, the highest average plant height was 71 centimeters, and highest average stem thickness was 6.04 millimeters in the control plants, and the highest number leaf was 17 in the AM6 line. Table 1, some characteristic control and mutant quinoa genotypes. You can see you can see table 1, highest plant height measured from control plants 71, lowest plant height MI4 60, 40, 60, plant stem thickness highest measured from M control plants 60.04, lowest plant stem thickness measured EM6 3778, leaf number highest EM6 17, lowest leaf number EM4. Table 2, seeds yield and some characteristic of control and mutant quinoa genotypes. You can see biological yield, highest M control plants 90.1, lowest biological verum EM4 68.0, seed yield gram plant, highest seed yield M control plants 6.74, lowest seed yield EM4 1.36, maturation time, longest maturation time 141 M control, between different control and mutant lines, important percent 5. You can see table 2. Conclusion, the results of the study revealed that the mutation had different effects on plant height, leaf number, and maturation time. It decreased biological yield and seed yield. The genotypes still showed erratic results, indicated that the settlement is not complete. Therefore, it would be useful to sow and spread this seed from this generation and carry out study one generation further. Thank you for your attention.
Thank you for your presentation. Question? There are some questions? No. Thank you again.
Thank you.
The next presentation and the last one of this session is TIFAR invasion and control in California rice. I invite Denise Enchi to do the presentation.
Good afternoon, everyone. I'm Deniz İnce. I'm a PhD candidate at UC Davis. It's my pleasure to be here today. I'm a wheat science person by training and primarily working on rice cropping system. Today I'm going to talk about an invasive weed and aquatic species, Typha latifolia. Before that, I would like to give some background information about California rice. It's the second largest rice producing state in the United States after Arkansas. We have approximately about 200 hectare rice production annually. Due to severe drought that was declined 50% last year in 2022, we got 3 years of drought consecutively. We hope to recover that in time, but that shows us that water management and drought is an ongoing problem, and it's growing in the valley. Nearly 97% of the rice is growing in Sacramento Valley, where you see on the map here, and we got some drill seeded rice in Sacramento San Joaquin Valley, where you see the red star. It's the capital, Sacramento, and on the left side, that's a drill seeded rice. It's different production than the water seeded, and that's why we have typha problem over there. So literature says wheats are capable to reduce 90% of the yield, but I can say it's easily 100% of the yield reduction if you don't have a chemical wheat management. And most rice production is heavily dependent on the chemical wheat management. Sacramento Valley, because of the clay soil type, has no crop rotation. All rice fields are rice every other year. So we also have a huge herbicide resistance problem. I'm going to talk about a new chemistry here, fluoropyroxyfenbenzyl. It's a synthetic auxin. We got this herbicide in 2018 in the south, but it took 5 more years to be able to get registration in California. This year was the first year that growers were able to test the herbicide. They got a good results. It mainly covers sedge species, broadleaves. Even though it's a synthetic auxin, it also has activity on some grass species. So we were happy with that. And the application window is from 2-leaf rice stage until the 60 days before harvest. So Typha latifolia, also known as common cattail, normally exists in the irrigation canals. drainage canals, ditches, lakes, marshlands, rivers, streams, but they normally don't exist in the rice fields. Here in the San Joaquin Delta, we have marshlands, and they are drained and then became rice fields. So therefore, we have the rhizomes in the soil. In 2022, we got too many calls from growers, and then we put a study about the efficacy of fluorpyrex fenbenzyle. We went with the 40 gram active ingredient per hectare rate, which is the label rate for other wheat species in rice. And then we went with the double rate, and then combined with triclopyr, which is also another synthetic auxin, and then triclopyr alone, of course. All of them included methylated seed oil 1% to increase the efficacy. So last year, we got a pretty promising results. If you look at the data columns here, at 28 days after treatments, we got very, very good control with up to 2 meters tall typha species. When the typha was less than 1 meter tall, we got 100% control. And then we shared this data with the other colleagues and collaborators, so we would like to repeat the study this year to get the registration on label against this wheat. And then in 2022, we added this new treatment list. So we have a timing of up to 1 meter tall typha and then 1 to 2 meters tall. We went with 40 gram AI in the first treatment, second treatment as well 40 gram, but now we have a late stage application. In the third one, we have a sequential application of 14 days apart, up to 1 meter and then 1 to 2 meters tall. And then we got 2x rate of 80 gram per hectare, up to 1 meter tall, and then fifth treatment up to 2 meters tall. And then the sixth treatment, we have a combination of triclopyr and fluorpyrex fenbenzyle, and then triclopyr alone, and then untreated check. So in the right side, you see the 3 meter by 3 meter plot that where I sprayed the herbicide. So on the sides, you see like typha, like a wall, then they are like 2 to 3 meters tall. And the efficacy is like 100% when the typha is 1 meter or less. On the left side, you see the 14 days after treatment injury on cattail, and then rice is pretty good standing. They don't have any injury. If you look at the control data here, for the listed treatments, except the triclopyr alone, all of the treatments got 100% control at 28 days after treatment. Triclopyr doesn't have activity on cattail, but when you combine with fluorpyrex fenbenzyle, you get a faster control, maybe in 2-3 days instead of 14 days. Right now we are working on the molecular analysis of why the combination increase the activity but doesn't change the total results at the end. Here we have the height of treatments at 28 days after applications. You see the control on the 8th column is almost 2 meters tall, and the other treatments decrease like almost up to 50 centimeter tall typha, except the 7th treatment, which is triclopyr alone. I have 2 columns missing here because they were too late application, up to 2 meters. Here I have those data. The decline is more than 50% versus the untreated check. Here I have a triclopyr applied plot in that picture. You see that there is no efficacy, no injury at all. That's my dry weight data. On the eighth column, I have untreated check around 50 gram per plant. But on the other side, even with the triclopyr, I don't have any control or efficacy, but there's a decline in the dry weight. We shared the data with the responsible colleagues to get the label, and looks like in a few years we will be able to get a temporary permit to apply against cattail. With that, I would like to conclude and thank my colleagues on the extension side, Michelle, Leinfelder, Miles, Rice Grover, Ben, Jeff, and Zuckerman families, and the funding resources. If you have any questions, I would like to answer.
Thank you for your presentation. Question? No question.
Thank you.
Thank you again. So this was the last presentation from this session. So we will take the break sometime before the time for break, because the next session will start at 3:30 p.m.
6th session of the Congress. The first presenter is the Professor Maria. He's going to discuss about the sunflower hybrids with high-speed yield potential and high oil quality for human health and food security. Welcome, Professor Maria.
Okay. My name is Maria Păcureanu. I work as sunflower breeder at the biggest institute for the field crops in Romania, near Bucharest. Also, I work in a center for biodiversity in Romanian Academy. I will do a presentation about the importance of obtaining sunflower hybrid for which to have the characteristics which are for which there is a request in the actual seed market. Sunflower is an important crop of the world, having the fourth position after palm, soybean, and oil rape. The total cultivated area with sunflower over the world, it's around 29 million hectares, with more than 50 million tons seed yield per year. The European Union countries have the third position regarding the area cultivated with sunflower after Ukraine and Russia. All Europe has 71% from the total world seed yield. This was a communication from 2021 by Etienne Piloche in France. Romania is the country with the largest cultivated area in European Union countries. Sunflower, what for is cultivated? Sunflower is cultivated first for oil content and oil quality. There is another sunflower type which is cultivated for seed, for eating seed or for using them in producing some sweets. This is a confectionery type of sunflower, and there is a type with high oleic acid content, this giving to oil high quality. To improve genetic progress in sunflower, it is a priority to improve competitiveness of this crop with other crops and to improve stability of yield and quality. As I told, there are two types of sunflower. The type for oil, it's high cultivated by farmers, and the confectionery type it's, has a small percent of the seed market. A sunflower seed may help, they are very important for the human health. They may help lower blood pressure, cholesterol, lower cholesterol and blood sugar, because they contain E vitamin, magnesium, protein, linoleic, good fatty acids, and other plant compounds. The new type of sunflower oil with high quality can have high oleic acid content, more than 85%, high palmitic, high stearic, they can be more than 25%, and good content in tocopherol. This give a good storability of oil and more healthy oil. Even in sunflower breeding, it was has been done effort in all breeding program for improving different characteristics. The seed yield which are obtained in farm are not at the genetic potential of the hybrid. So this is because of some limitative factors, biotic or abiotic, or because of some wrong technologies. Sunflower breeding activity must be intensified by creating a genotype which must have different vegetation period, different maturity groups, also to be resistant to drought and to high temperature, air temperature, especially in flowering time, tolerant to salinity, tolerant to low temperature in germination emergence period. If we compare sunflower with other crops cultivated in zones with temperate climate, sunflower is a recent crop and has a narrow genetic base, but fortunately sunflower has many wild relatives, and this can help cultivate sunflower. Using wild helianthus species, it is a long tradition in sunflower breeding, and by creating interspecific hybrid, it could be obtaining many inbred lines with good characteristics, and all this line together with some wild population represent a very important genetic resource for increasing genetic variability in cultivated sunflower. The main trait for which it is made breeding, there are seed yield and yield components, high oil content, high oil quality, resistance to diseases, resistance to broom rape. It is an important parasite which attacks sunflower, resistant to drought, resistant to herbicide. It is possible to develop hybrid with genetic potential to 6 tons per hectare regarding the seed yield and oil content of 55%. But in farms, there are not obtaining such result. Even the new hybrid have very high potential, genetic potential. This is because, as I told, maybe because of some technologies and because of other factors. Oil quality, the breeding for oil quality, it's done now, it focuses on high oleic acid content, also high palmitic, high stearic, and the good tocopherol, especially alpha tocopherol in our breeding program. Here are some results regarding the genotypes. The analysis of fatty acid content. We succeeded to obtain some hybrid which more than 86% of high oleic acid, and with linoleic acid content more than 75. This is a conventional type of sunflower having high linoleic acid content, and for the genotype which have very low linoleic acid content, these are the genotype which have very high oleic acid content. Also, in our breeding program, we succeeded to obtain some genotypes which have around 500 milligram per kilogram oil of alpha tocopherol, but in this direction it could be made much more, because to have to 1,000 milligram per kilogram of oil, alpha tocopherol. Sunflower tolerance to herbicide has increased competitiveness of this crop because of identifying also one wild helianthus species, which has a gene for resistance to two types of herbicide, imidazolinon based on imazamox and sulfonylurea, which is the tribenuron. Using this type of genes, it was created, there have been created two systems, clear field system and express sun system for cultivating sunflower, and in present all seed market over the world is focused especially on hybrid resistance to herbicide. Yeah, this I told you about. And these are some images which sunflower sensitive to herbicide and sunflower resistant to herbicide. Using the sources created from these wild species for resistance to herbicide, it has been obtained many lines resistant to one type or to other type of herbicide, but in present there is activity for obtaining hybrid lines having the AIR gene. This gene was created by a company in Idera in Argentina in collaboration with BASF company, but now this gene belongs to Syngenta company. And we obtained this new gene by contract from Syngenta and put in our breeding program. Resistance to diseases is an important limiting factor for sunflower production, and it's very difficult to keep the control of some pathogens because of changing in racial composition very fast. And for this reason, it's necessary to do a good management of genes for resistance, also to do the monitoring interaction pathogen host plant. As I told you, sunflower, it's attacked by one parasitic weed, Orobanche cumana, broom rape, and it was done much work over the world for creating resistant or tolerant genotypes to this parasite, and even in all the breeding programs, there have been obtaining good results, good hybrid resistant to this parasite, but the parasite is changing very fast the virulence of races, and for this reason, the breeders and geneticists are looking all the time for the new sources of resistance. As I told, sunflower, cultivated sunflower has many wild relatives, and we tested, we are testing all the time, each year, some wild species for resistance to some pathogen or to the broom rape parasite. And in this result, I present you some result obtained for resistance to the pathogen Plasmopara hastede, which produced down a mildew. We identified 9 wild species full resistant to this pathogen. For parasite broom rape, we identify 5 species, but only 2 species are resistant to both pathogen and parasite. It is the number 10 and the number 16, Helianthus debilis and Helianthus pauciflorus. These 2 species, wild species, have resistance to both pathogen and parasite, and they are very important for us, for using in improving resistance in cultivated sunflower. For drought resistance, also we are using different wild species, but the best one until now in our program, it was the Helianthus argophilus. Here in this picture, we see near cultivated sunflower, and This wild species is very important, having good characteristics which confer resistance to drought. In our breeding program, in the last years, we obtained good genetic progress because we obtained the registration in the official list different hybrids. All of them are resistant to one or other type of herbicide, and some hybrid has good seed yield potential, more than 4 tons per hectare. Also good high oil content, 50 to 53%. For the next future, the direction at global level for sunflower crop are some direction. For sunflower competitiveness, it is necessary to create genotype well adapted to different environmental conditions because of climate change. Even sunflower is a crop with some tolerance to drought, but in the condition of climate change, there are problems. In the last years, there have been problems with sunflower in different areas. To create genotypes having characteristics which are requested by seed market, creating genotypes which have the breedability for cultivation in different technology systems. To the request for some morphological and physiological characteristics is to have tolerant to high temperature, also to water missing in time of pollination period, tolerant to low temperature in germination emergence period, tolerant to frost, to have the capacity to recover after hail, because we have such hybrid, good efficiency for the water utilization. And regarding the technology, to cultivate genotypes or to have some technology which can have drought escape. This it means that we can planting in early period, to have some genotypes with short vegetation period, also to practice intercropping cultivation system, minimum tillage system, cultivation in the forest intercropping system, cultivation with low cost in the ecological system, friendly, we have technology, genotypes and technology friendly with pollinators, because it is important for pollinators, for human also. Good strategy of vegetable rest utilization with allelopathic role in the wheat control. And thank you for your attention.
Thank you, Professor. Okay, any questions? Okay, can I ask one question from you? Okay, what about the situation of the sunflower in your country, whether it's grown once in a year or twice in a year? Once in a year? Only one time in a year? Okay. Because from where I am basically from Pakistan, we grow sunflower twice a year. One in the autumn season, it's usually grown in October, and the other one in the spring season. It's a winter. So then you can grow 2 crops of the sunflower in a year. Okay, and why you people are not switching towards the genotypes instead of the hybrids? Due to their yield? Okay, thank you. So the next presenter is Alp Ayan. I don't know how to pronounce it. Sorry for that one. And he's going to present his research work on the proteomic evaluation of multiple abiotic stress-tolerant soybean mutants.
Good afternoon, everyone. Before I begin my speech, I want to thank to the organization committee for this good opportunity, scientific opportunity for us, and they are increasing their numbers every year, a successful organization. I want to thank all the organization committee and all the other personnel who work for this organization. This is my second participation to this Congress. Last year we presented RNA sequencing study of our research. This year we took our research to the next level. Last year it was a transcriptomic research. This year we evaluated our mutants with proteomic analysis, two-dimensional proteomic analysis. So it's part of the previous project. I'm from Istanbul Kültür University. I'm working as assistant professor there. With our research team, we are working on plant biotechnology basically. And in plant biotechnology, it's a very large topic, of course, but we are working basically for transcriptomic and proteomic analysis and also mutation breeding studies. This is the part of our latest mutation breeding study. So if I start, our aim was to inducing random mutations by gamma radiation in soybean plant. Professor Maria referred to soybean as one of the 4 major oil crop plants with, of course, sunflower and some other plants. We are working also with an oil plant, it's soybean. So we are trying to develop new varieties, new mutants, which are tolerant to multiple stress factors as draw stress and salinity stress. Second aim was, actually I mentioned the second aim. The third aim is confirmation of tolerance through some selections because as we all know, gamma radiation and all the mutagens create random mutations on plants. So after creating large number of mutants, we have to select the beneficial mutants among all the population. So the third aim was confirming the tolerance for our case, it's salinity and draw stress. Our fourth aim was determining genomic, transcriptomic, and proteomic alterations in the mutant plants. So I'm going to talk about the last part of this aim. It's the proteomic part. I want to summarize the whole process for mutation breeding first. In plant biotechnology, we can try 2 different approaches basically. One of them is forward genetic approach. The second one is reverse genetic approach. Actually,
they are differing by their mechanism. In forward genetic research, you create some kind of mutant phenotype. It can be created by physical mutagens or some chemical mutagens, or lately in literature we can also see some biological mutagens. Some researchers identify transposons as the biological mutagens, but we are using a classical physical mutagen, gamma radiation. After the creating mutant phenotypes, you have to identify the mutant alleles, you have to identify mutant genes. After that, you have to do some transcriptomic research if you want to identify what genes are expressing differently. And the last part, of course, after transcription, you have to identify your mutants in proteomic. The last product is always proteins, or some kind of different molecules also can be, but the proteins are the major group. So you have to evaluate your mutants in proteomic way. The reverse genetic approach is more popular lately, but we are not doing this right now. As we all know, it's opposite the random mutations. You create targeted mutations by gene transfer, or lately CRISPR or different kind of gene editing techniques can be applied. But today I'm going to talk about forward genetic approach. In material methods, we irradiated like 12,000 seeds, soybean seeds, in different doses, because you have to find the efficient radiation dose first, because higher radiation doses can be lethal, of course. The lower radiation doses cannot create the efficient mutation rate. So first we have to find the efficient radiation dose to use. For this purpose, usually researchers use the dose that reduce the growth of plant like 30% or 50%. In our case, we used the radiation dose that decreased the plant growth 30%. So this dose was 250 grays of gamma radiation in our case. So after determining that dose, we irradiated our seeds, as you can see in this second picture on the upper part. After that, we planted all these irradiated seeds to the field. Actually, this project was conducted with the help of Black Sea Agricultural Research Institute in upper southern, northern part of Turkey. These fields belong to them in Samsun, the city, Samsun city in Turkey. After the plantation of these seeds, we did very extensive selection applications, because we got lots of seeds from all these 12,000 plants. After that, first we did our selections for the salinity tolerance. We applied 120 millimolars of sodium chloride to our mutant plants. After that, we obtained around 12 mutants, 12 salinity tolerant mutants. Among these mutants, we did the secondary selection for drought tolerance also, because as I mentioned earlier, gamma radiation creates random mutations, and the gamma rays can change multiple targets in your genome. So some targets can achieve salinity tolerance, and some targets may also create drought tolerance as well. So we were a little bit lucky actually, because in this kind of study, if you irradiate 12,000 seeds, you can get roughly 2 or 3 beneficial mutants. In our case, we got 4 multiple stress tolerant soybean mutants. Then Of course, we wanted to confirm the tolerance in vitro conditions also, because sometimes in vivo conditions can give wrong results. Because of that, we always want to confirm our tolerance in both ways, in vivo and in vitro. After the in vitro confirmation, as you can see in the bottom left corner, we got 4 salinity and drought-tolerant mutant plants, and we conducted our two-dimensional proteomic analysis on these 4 tolerant mutants. In two-dimensional proteomic research, I want to summarize the workflow for this kind of research first. The protein, of course, the sample collection is a vital part. After that, protein extraction and purification parts all are the major parts, because if your proteins are not purified enough, some plant phenolic compounds or some plant pigments can block your proteins to run in the gels. So the protein extraction and purification parts are very important in this kind of research. For this purpose, of course, we can use different kind of kits manufactured from different companies, but in kit isolations, in plant samples, protein concentrations are not very high, and in this kind of research you need at least 200 micrograms of protein to be separated in the polyacrylamide gels. So we are using more manual approach. It's actually working very well in plants. It's a trichloroacetic acid acetone approach. It's manual approach. As we all know, acetone dissolves the plant pigments very well, so we can remove them from our extract easily. Trichloroacetic acid working in both ways. It helps to damage and demolish the hard cell wall of the plant cells. Also, it blocks the protease activity in the samples, so it protects our proteins. After that, purification can be conducted by any kit. It's easier after the good extraction protocol. After that, you can either separate your proteins as fractions, or you can digest them by, for example, trypsin, and you can also separate your peptides differently. And there are different options. As DIGE, you can attach some kind of fluorescent dyes to your proteins, and you can analyze them differently. Also, iTrack is also popular lately. Or besides all these procedures, you can conduct two-dimensional separation. First dimension is according to proteins isoelectric focusing values. We can call them pH values. The second separation is based on proteins molecular weight. So this two-dimensional separation helping us to identify our proteins very easily. After that, we need to identify our proteins in different databases, so we need to use mass spectroscopy to identify all the protein spots. When we get the raw data from the mass spectroscopy, the bioinformatic part starts. After that, I'm going to present the results of KEGG analysis, string analysis, in the further slides. Before all these, we applied draw and salinity stress to our soybean plants, and we wanted to show the indication of stress first on the plants. For this, of course, there are lots of different ways to show the indication of stress, like MDA analysis or some kind of other metabolites. But in this study, we used proline content and the total carbohydrate contents of the plant for this purpose. In this, graph, you can see the proline levels increased after the application of salinity stress. And also we used in vitro draw application in this part. We used polyethyl glycol for the draw application, and in both salinity and draw applications, proline levels increased. Also, we used total carbohydrate content of plants as the indicator of stress. As you can see here, all the carbohydrate levels are increased after the draw and salinity stress. Actually, both salinity stress and draw stress are osmotic stresses, and we know that the osmoprotectant molecules as different sugars and proline as amino acid increase as the reaction to the both osmotic stresses. So we tried to show the indication of stress, and we saw the indication from this chart. After that, as I said earlier, we separated proteins in 2 dimensions. First dimension is based on the proteins' isoelectric focusing values. As I said, it's pH values of proteins. These gels, in this first separation, we have different options. If you are targeting the specific proteins, you can use more narrow range of separation as pH 5 to 8 or 4 to 7. If you are sure that your targeted proteins are not separated in lower pH or higher pH, you can use more specific IPG strips. But in our case, we wanted to see whole proteome profile, so we used IPG strips starting from the pH 3 to pH 10 in plants, and mostly in most of the eukaryotic organisms. Below 3 pH and above 10 pH, you cannot see too many proteins. So these are IPG strips, I mean from pH 3 to pH 10 is very useful for our research. In these gel images, you can see there are lots of protein spots. All these dots are different proteins. And in each gel, we had like around 800 to 900 protein spots. Of course, we cannot select all the differentially expressed proteins by our bare eyes, so we are using some kind of softwares. We are using Biorat software, PDQuest software. This software analyzes all these images, and we are determining some thresholds for the program. In our case, we are using 2-fold increase or 2-fold decrease threshold. You can use higher thresholds like 4 times, 5 times, 3-fold, 4-fold or 5-fold based on your research, but it decreased the amount of differentially expressed protein counts, and you can easily miss some proteins through your analysis. So we are using a lower threshold, like 2-fold, and it gives us around 55 differentially expressed proteins after the draw and salinity stresses. Among these 55 proteins, this is the system that we used for the analysis of the spots. We are getting our raw data by this device. After this device analyzes the spots, you can create different charts. First of these charts was the cellular localization of differentially expressed proteins. In our mutants, most of the differentially expressed proteins were belonging to chloroplast, so we were expecting to we were expecting to find a major differences in photosynthesis, of course. The second biggest part was the proteins in the cytosol. And you can see there are some minor changes in mitochondria, cell wall, cell membrane, nuclear proteins, chloroplast, and mitochondria also. and some other various proteins. After that, we evaluated the cellular functions of these differentially expressed proteins, and as you can see, the higher ratios were also belonging to photosynthesis, like 15%, and after that, the photosynthesis process created changes in carbohydrate metabolism as well. We also saw 15% antioxidant enzyme regulation proteins in this differentially expressed proteins. And the lower parts, as you can see, amino acid metabolism is affected. ATP synthesis is also affected as 9%. After this analysis, it gave us a vision. We understood that our gamma radiation application changed basically photosynthesis and the carbohydrate metabolism in our plants, and these differences gave tolerance capacity to our plants against salinity and draw stresses. After that, also that raw data analysis that I mentioned earlier gave us the accession numbers of all these proteins, 55 different proteins I said. In string analysis, this is a free software in internet, you can use the accession number of your proteins, and it gives you the protein-protein interaction maps of the selected proteins. So when we evaluated all these 55 differentially expressed proteins, as we expected, we saw that the major groups were like these green dots belong to photosynthesis mechanism, and the red dots belong to carbohydrate metabolism. So as we saw from the previous charts, we also showed that protein-protein interactions are also mostly changed in photosynthesis and carbohydrate metabolism in our mutant plants. There are some blue dots. As you can see, they are not interacted with other mechanisms, so they are minor changes which also contribute to the tolerance, but they are not so major. After that, we also created cake pathway analysis by these accession numbers, and it gives us the localization of the proteins in the metabolic pathways in plant cells. As you can see, I'm not sure if the colors are visible from where you're watching, but the green, do I have a cursor here? Or I can... Synthesis. Actually, after that, we also evaluated the carbohydrate metabolism in the plant system. Especially pentose phosphate pathway is fortified by our proteins. As you can see, green boxes are much more around here. So we changed lots of different proteins in the pentose phosphate metabolic pathway in the mutant plants, in our mutant plants. So these two machinery, in our research, we found that these two machineries are the most major improvements in our plants. As conclusion, we can say that the gamma-induced salinity and drought-tolerant soybean mutants were obtained, as I showed earlier. In these two machinery, I mean in photosynthesis and in pentose phosphate pathway, especially glyceraldehyde 3-phosphate dehydrogenase enzyme is changed significantly, contributed to salinity tolerance, and phosphoribulose kinase contributed to salinity tolerance of our mutant plants also. and carbonic anhydrase enzyme also contributed to the salinity tolerance of our mutant plants. The most significant protein which contributed to draw stress was fructose biphosphate aldolase enzyme in our mutant plants. As I mentioned earlier, our first selection target was salinity tolerance. So as you can see, the protein's salinity tolerance contribution was more than draw tolerance. The second selection was for the draw stress tolerance. So we changed, actually, probably we changed much more proteins for draw tolerance, but it was our secondary selection target. So probably we eliminated some draw tolerant mutants in our previous salinity selection. So from this part, we want to take these mutant plants for further analysis and register them for as new varieties. We are working on this part in our project. This study was also financially supported by the Republic of Turkey Ministry of Agriculture and Forestry General Directorate of Agricultural Research and Policies by the grant number of TAGEM 17 RG 56. And this project was finalized 2 years before, but we are still working on the molecular parts of the this project. This is the small part of our team, but the major part, I can say. We also have some undergraduate and graduate students, but when we were taking this picture, they were not around, so they missed the chance. Thank you for your attention. If you have some questions, I would like to answer them. It's coming now? Oh, that's good.
No issue. It's not a question, it's some sort of discussion. You have measured the, can you go to the previous slide? Okay, thank you. Our next speaker is Tahir Benzina Farida. Is she here? Okay, the next one is the Jurmen Sari. He's going to present his research work on the title Impact of AMPEP concentration in nutrient medium on the growth and lipid accumulation of cholera species culture.
Hello. Ladies and gentlemen, greetings. Allow me to introduce myself. I am Jurmen Etsari from the Philippines and currently studying at Kastamonu University, Turkey. It is my pleasure to present to you my topic entitled Impact of AMPEP Concentration in Nutrient Medium on the Growth and Lipid Accumulation of Microalga chlorella species culture. This will be the overview of this presentation. Introduction. Microalgae are photosynthetic, prokaryotic, and eukaryotic microorganisms that can proliferate, making them potential renewable nutrition sources, biodiesel producers, and eco-friendly environmental alternative. Microalgae are nutrient-dense because of their biochemical composition, particularly lipid and fatty acid composition. The use of microalgae as direct feed in larval tanks is essential in aquaculture for bivalves at all stages, for some crustaceans and fish species larvae. Additionally, microalgae industry produces a variety of biotechnological products. This includes health food, natural colorant, and cosmetics. It can also be used as fertilizer. For example, microalgae used as nutrient enrichment for the production of agricultural product or seaweed farming, of course, to increase production. It can also be used as wastewater treatment. For instance, removal of coliform bacteria, reducing the both chemical and biochemical oxygen demand, and removal of some heavy metals. Also, it can also be used as pharmaceutical products or nutraceutical products. For example, supplemental feeds or supplemental food. Additionally, also used as bioenergy, such that lipid production or biodiesel producers or production. Of course, feeds in aquaculture environment. One of the microalgae species is chlorella species. Chlorella species is widely produced microalgae due to its high productivity, cellular composition, and ability to grow photoautotrophically, heterotrophically, and mixotrophically. Studies stated that chlorella species rich in pigments, fatty acid, lipids, protein, minerals, and vitamins. Furthermore, allow me to introduce this species, a Ascophyllum nodosum. Ascophyllum nodosum is a brown seaweed containing bioactive substances that may influence the molecular, biochemical, and physiological function of crop plants. And there are a number of temperate regions around the world, such as United Kingdom, Iceland, Ireland, Norway, France, and Canada, where this species thrives abundantly. And there is an extract from this species known as ascopylum, or Acadian marine plant extract powder, or AMPEP. This AMPEP or extract is widely produced or applied to increase the performance of land crops and has been reported to increase the growth of some seaweed crops, particularly either field cultivation or micropropagation of seaweed or macroalgae. However, the use of AMPEP on the production of microalgae is not yet conducted. Thus, this study investigates the use of AMPEP concentration for the production of microalgae, particularly chlorella species, of which we investigate the growth and lipid accumulation of chlorella species culture. For the materials and methods, microalgae and the culture condition, the microalga chlorella species obtained from the aquaculture department, Faculty of Fisheries, Kastamonu University, Turkey. And the AMPEP, or the extract AMPEP, was obtained from MSUTCTO, or Mindanao State University, Tawi-Tawi College of Technology and Oceanography, Tawi-Tawi, Philippines. And we use 500 ml flat bottom plus in triplicate with VG11 medium as nutrient medium. And there are three concentration group. Group A, these are the concentration for the AMPEP. Group A, the lower concentration. B, higher concentration of AMPEP. And C is the control group. And each solution was autoclave for 20 minutes at 121 degrees centigrade. These are the culture experiment. And the initial density of chlorella species is 1 million cells per ml. And artificial lightening in the laboratory, it was cultured in an artificial environment. And fluorescent lamp were used for lightening. It was cultivated in an air condition maintained at temperatures of 20 degrees centigrade. For the growth and lipid analysis, every 3 days of sample of microalgae were collected for cell counting and analyzing, of which we use hemocytometer for the cell counting under the light microscope. Then we also estimate the dry weight of chlorella species culture. Then the lipid accumulation was analyzed using a spectrophotometric method, was followed by the method of Misha et al. 2014. These are the culture experiments. For the statistical analysis, we use IBM SPSS software version 20 to analyze the data, growth, and lipid accumulation of chlorella species culture. And the mean values were analyzed by analysis of variance at significant level of 0.05. And Duncan test was used to rank the mean. This figure shows the cell number of chlorella species culture at different concentration of AMPEP. A represent the lower concentration of AMPEP. B, higher concentration in C, and C is the control group. According to the result of this study, AMPEP addition to the culture medium of chlorella species culture provides higher cell densities, of which lower concentration of AMPEP reach a cell density of 148 million cells per ml, which is significantly different than the higher concentration of AMPEP and the control group. In other study, using different concentrations of phosphate in a nutrient medium on Chlorella vulgaris culture obtained a cell density of 2.28 million cell per ml, which is lower than the present study. Another study, which is the effect of phosphate source in a nutrient medium, which of 32 cells for Chlorella species culture. Thus, the study presents lower concentration of AMPEP in a culture medium positively increase the cell density of Chlorella species culture. Moreover, this figure shows the dry weight of Chlorella species culture at different concentration of AMPEP. The yield of Chlorella cell cultures with AMPEP was estimated by measuring the Chlorella species biomass, or the dry weight, which, based on the result of this study, lower concentration of AMPEP produced higher dry weight than higher concentration of AMPEP and the control group. There was a significant amount of dry weight generated in the present study compared to the amount in other study. For example, the microalga nanochloropsis was cultivated in a flat photobioreactor with a high nitrogen and phosphate concentration in F2 medium and yielded a dry weight of 2.37 gram per liter. Another study by Feng et al. 2020, which Chlorella zingengiensis was cultivated on VG11 medium, in which with high nitrogen and phosphate achieved a dry weight of 0.90 gram per liter. The results of their study were comparable to those of the present study, of which lower levels of AMPEP in a nutrient medium lead to an increase in dry weight in Chlorella species culture. Thus, using VG11 medium with addition of AMPEP increased dry weight of Chlorella species culture. Also, this figure shows the lipid accumulation of Chlorella species. And the present study revealed that incorporation of AMPEP in the nutrient medium increased the lipid accumulation of Chlorella species than the control group. In other study, the Chlorella vulgaris was cultivated in an F2 medium, achieved a lipid content of 7.80. Comparatively, it is lower than the present study, with higher and lower concentration achieved. Higher lipid content of Chlorella species culture. Moreover, studies have shown that nutrient-limited can result high lipid content but low biomass level. For instance, it has been revealed that low phosphate concentration in VG11 medium lead to greater lipid accumulation of microalga Chlorella perinodusa. In the present study, high levels of AMPEP in a VG11 medium increased lipid accumulation of Chlorella species culture. To sum up of this study, a lower concentration of AMPEP in a nutrient medium significantly improves the growth of Chlorella species culture, particularly the cell density and the dry weight. Additionally, both concentration of AMPEP, either higher and lower in a nutrient medium, increase lipid accumulation. However, this study requires further analysis, such as other biochemical composition, such as pigment, fatty acid, protein, and etc. Also, to optimize the AMPEP concentration in the production of microalga chlorella species culture. These are the references used in this study. Thank you for your listening.
Any questions from the audience? Thank you.
Thank you very much.
The next presenter is Anreda Embrek. I don't know how to pronounce it. Sorry for that one. And she's going to deliver his talk on the effect of Moringa oleifera ethanolic extract on growth and hepta, stomatic induce of Nile telapa.
A pleasant afternoon to each and everyone. Before I'm going to present my topic, I'd like to introduce myself first. My name is Inraida Imbuk. I am from Philippines, but I am currently studying at Kastamonu University, Turkey. So my topic to be presented today entitled Effects of Moringa oleifera ethanolic extract on growth, viscerosomatic and hepatosomatic indices of Nile tilapia or Oreochromis niloticus. So the overview of my presentation are introduction, materials and methods, results and discussion, conclusion, and references. So for introduction, aquaculture has recently expanded due to the growing demand for sustainable animal protein to feed the world's populations. The Food and Agriculture Organization, or FAO, estimated a total of 122.6 million tons of aquaculture produced globally in 2020, generating US dollar $264.8 billion. Additionally, aquaculture production contributed a record of 49.2% of the world's total productions of aquatic animals in the same year. Tilapia, belonging to the family Cichlidae, is one of the most common farm fish worldwide and is regarded as the second most significant freshwater fish. They are generally endemic in Africa's water, but have been introduced into many tropical, subtropical, and temperate regions. Tilapia productions contribute substantially to job employment, income, and food security. Furthermore, tilapia is an excellent choice for aquaculture due to its fast growth, adaptability to various environmental conditions, capacity for in vitro fertilizations, ability to feed on low trophic levels, and disease resistance. The rapid growth of modern aquaculture is driven by a variety of factors, which include the increased use of formulated aqua feeds and intensification of culture systems. However, efforts to intensify aquatic animal species can lead to increased diseases, limited growth performance, and stress. For a long time, antibiotics have often been used in aquaculture industry, not only to prevent or control infectious diseases caused by microbial pathogens, but also as a growth promoters. However, applying antibiotics as a growth enhancer in aquaculture is increasingly expensive and environmental and hazardous problems. Hence, alternative medicinal plants have received more attention to substitute expensive growth in stimulant and prevent potential health and environmental risks. In fact, herbal medicines and their extract have been proven to enhance aquatic animal species growth, antioxidative, and stimulate immune responses. So medicinal plants are cheap and easy to prepare and effective with fewer side effects during the treatment of diseases and without any environmental and hazardous problems. So medicinal plants include herbs, spices, seaweeds, herbal extracted compounds, traditional Chinese medicines, and commercial plant-derived products. So Moringa oleifera is one of the medicinal plants that has many health benefits and nutritional properties. These plants belong to the family Moringaceae, and it is widely distributed worldwide, particularly in Asian and African countries. So this plant is considered as one of the most valuable trees since almost every part of this plant can be used for food, industrial purposes, and medication. So Moringa oleifera can be used as food for human consumption, and it can be utilized as livestock feeds for animals. So this plant holds considerable potential for feed ingredients due to their high nutritional quality, including amino acids, fatty acids, vitamins, and minerals. Moringa oleifera is a good source of protein. In fact, 260 grams of crude protein, of which about 87% is a true protein. So the increasing commercial value of the Moringa oleifera plant proved to be an excellent source of phytonutrients and has nutritional and medicinal applications for aquatic animals. It is a cost-effective, easily procurable, and rich source of multivitamins. Hence, the objective of this study is to evaluate the dietary supplementation of Moringa oleifera ethanolic extract on growth performance, viscerosomatic, and hepatosomatic indices of Nile tilapia. So for the materials and methods, the experiment was designed as fully randomized. The fish with an average body weight of 10.05, positive negative 0.87 were purchased from Adana, Turkey, and they were transported to Laboratory Aquarium, the Faculty of Fisheries, Kastamonu University. A total of 120 fish were stuck in a 12-mesh cage, and all tanks were supplied with aerated and at 27 degrees Celsius water temperature. Prior to the feeding trial, fish fed twice a day with the commercial diets ad libitum by hand. So for the extraction process of Moringa oleifera, the leaves were collected from Indonesia, then dried at room temperature. After the drying process, it was grounded with high-speed mill. Then, for the preparation of a chaos ethanolic extract, we use 100-gram moringa powdered with 200 ml ethanol, and we kept at room temperature for 72 hours. The extract was filtered through a plastic nylon mesh strainer, then evaporated in a rotary evaporator at 80 degrees Celsius. So the exact amount of extracted solution was added to the experimental diets in accordance with the given doses. So for the preparation of feeds, the moringa extract was added to the fish basal diet at 0, 1%, 2%, and 3% through spray method. For sampling was done every 2 weeks throughout this study. The fish were starved for 24 hours, then anesthetized by phenoxethanol. Then fish were individually taken in weight and length to determine the growth performance parameters. And liver and viscera were measured to determine the viscerosomatic index and hepatosomatic index. So for the growth parameters of Nile tilapia was measured using indices such as weight gain, specific growth rate, and feed efficiency ratio according to the formulas given by Ricker 1979. So for the computation of weight gain final weight minus initial weight divided by initial weight, and for the following computation. For the viscerosomatic and hepatosomatics, liver weight divided by total body weight times 100, and for visceral weight divided by body weight times 100. So for statistical analysis, one-way analysis was done using one-way analysis of variance ANOVA with the help of IBM SPSS software version 20 with level of significance 0.05. So the result and discussion. The growth, performance, and feed utilization are given in this table. So the obtained results of the growth revealed that the final weight gain, specific growth rate, and feed efficiency ratio was significantly higher by adding moringa extract on the diet of Nile tilapia for 75 days cultured period, in which the highest final weight gain, SGR and FER, were observed in fish fed with 1% moringa extract or MO1 compared to the other experimental groups. So the improved growth, performance, and feed utilizations are important indicators for evaluating the effects of medicinal plants in aqua feeds. So in the present study, dietary incorporation at 1% moringa ethanolic extract significantly improved the growth performance in feed utilization of Nile tilapia. Similar finding has been reported that moringa leaf meal at 100 gram demonstrated better growth performance and feed utilizations of Bocart's catfish or Pangasius bocarti. In addition, an inclusion rate of 0.5% moringa leaf extract could increase the growth performance of freshwater-prone Macrobranchium rosenbergii. Previous studies has been reported that the moringa leaf at 0.5 and 2.5 enhance the growth performance of white-leg shrimp Penaeus vanami. So according to the results of the previous study, the increased growth rate and improved feed utilization can be linked to immune system properties in high digestibility, absorption and assimilation through enhancement of gastrointestinal enzymes and intestinal microflora due to Moringa oleifera prebiotic activity. In addition, the improved growth performance might be due to the high concentration of antioxidants contained in this plant, such as B-carotene, vitamin C, and quercetin, which improve the health conditions of fish. So in the present study also showed adverse effect on the growth of Nile tilapia fed with the basal diets containing 2% and 3% moringa extract. But according to Poicha et al. 2017, the dietary moringa species should be considered in the diet of P. bocarti in such a way that the amount was not over 100 gram fish. So the non-significance of Moringa oleifera on the growth of Nile tilapia could be attributed to antinutrients contained in this plant, such as phenol, tannins, phytates, saponins, glucosinolates, non-starch polysaccharides, protease inhibitors, and possipols. So these are the ingredients which have a bitter taste that may cause low acceptability of feed to the experimental fish. So this figure is the result of viscerosomatic index of Nile tilapia fed with different levels of Moringa oleifera ethanolic extract for 75 days. So bars with the same letters are significant, while bars with different letters are significant. So the viscerosomatic index of Nile tilapia fed with 2% obtained 19.27, positive negative 1.17, which was significantly higher than fish fed with 1%, which is 15.51, positive negative 0.95. In addition, the hepatosomatic index of Nile tilapia fed with 2% was significantly higher, which obtained 4.89 positive 0.6 compared to the control group. So the assessment of nutritional utilization and biochemical composition in the feedstuff can be carried out using morphometric characterization of the fish, such as an organ and tissue indices. So in the present study, a 2% moringa extract inclusion significantly increased the viscerosomatic index and hepatosomatic index of Nile tilapia. So there are studies use variety of medicinal plants for the improvements of organ indices of different species of fish. For example, the lemna minor leaf meal at 25%, 50%, 75%, and 100% as a dietary supplementation significantly increased the viscerosomatic and hepatosomatic indices of Clarias garipinos. Another study has been reported that the maltose as basal additive to fish diet increased and improved the condition of fish in terms of viscerosomatic and hepatosomatic indices, which an indication of the healthy with moderate energy reserve status of fish in the experimental diet than the control group. So according to Songbesan et al. 2017, the corresponding increase in weight of organs in tissue is due to the bioactive compounds in the additives. Another study has been reported that Jatropha coccus kernel meal used for feeding common carp fingerlings, higher value of hepatosomatic indices was observed in plant protein feed groups. So it could be suggested as a result of higher lipid deposition in the liver. However, although it has been reported that moringa leaf has low lipid content, which was attributed to the potentiality of moringa leaf to reduce cholesterol. Thus, the higher inclusion level of moringa extract in each fraction might therefore reduce the energy available for protein in lipid biosynthesis and led to lower growth performance and nutrient utilization. So in the present study, it could be said that the highest value of organ indices observed in groups with higher level of moringa extract seem to be as a result of low body mass due to lipid in the flesh. So in our study, we conclude that the effects of moringa oleifera supplemented tended to be dosage dependent, whereby the growth performance of Nile tilapia was enhanced as the concentration of moringa oleifera in feed increased from 0 to 1%, but the growth declined when the concentration was increased to 2% and 3%. So therefore, 1% of moringa extract added on the diets potentially enhanced the growth, performance, and improved the organ indices of Oreochromis niloticus or Nile tilapia. So these are the references. Thank you.
Thank you. Any questions from the audience? Okay, in the presentation, you have told that you collected the plant samples from the Malaysia. You collected the moringa leaves from the Malaysia and then imported or exported to here Turkey. How much it will take time? Because the literature says if we preserve the moringa leaves for a longer period of time, the efficacy of the moringa that starts declining. So the last presenter is will discuss about the sequence.
Hello everyone, I'm coming from the University of Çukurova, and today I will talk about sequence analysis of hydrolytic enzyme synthesis by rumen microorganisms. And what's rumen? Ruminant animals are herbivores that can utilize high amounts of plant cell walls and constitute a significant portion of all domestic animal species in the world. The digestive tract of mammalian herbivores, which use plant material as a diet, consists of four main parts: esophagus, stomach, small intestine, cecum, and large intestine, while the stomach of ruminant herbivores consists of four parts: rumen, reticulum, omasum, and abomasum. And what's the celluloses? Celluloses are multi-enzyme complexes consisting of three main components: endo-beta-1 4-glucanases, exo-beta-1 4-glucanase, or cellulobihydrolase, and beta-glucosidase. Some cellulose systems also contain exo-beta-1 4-D-glucan, 4-glycohydrolase, and exo-beta-1 4-cellulobiositase. These three components act synergistically to hydrolyze cellulose to glucose. And what's caseolinase? Caseolinase and beta-caseosidase are the main enzymes responsible for the hydrolysis of caseolin. Caseolinase cleaves the internal beta-1,4 links of the caseolin backbone. Beta-caseosidase facilitates the hydrolysis of caseolin by hydrolyzing caseobiose and small caseolo-oligosaccharides to caseolose. Material and methods: obtaining data from NCBI database with FASTA format. Data on hydrolytic enzyme genes were obtained by selecting nucleotide or protein from the preferences on the main screen of the NCBI database and entering the name of the microorganism to be investigated in the query section. The following figures show this query screen. Obtaining nucleotide and protein sequences of cellulose enzymes. Nucleotide and protein sequences related to cellulose enzymes were obtained by displaying the information of the microorganism to be investigated on the result screen and selection non-whole genome base pairs. The data were saved in FASTA format and stored in two separate folders. The path followed for downloading nucleotide sequence data in FASTA format in cent-coding sequences, FASTA nucleotide create file for protein sequences sent to file FASTA create file. and obtaining nucleotide and protein sequences of caselinase enzymes. Nucleotide and protein sequences of caselinase enzymes were obtained by displaying the information of the microorganism to be investigated on the result screen and selecting non-whole genome base pairs. The data were saved in FASTA format and stored in 2 separate folders. The path followed for downloading nucleotide sequence data in FASTA format is sent to coding sequences, FASTA nucleotide, create file. For protein sequences, sent to file, FASTA, and create file. Multiple sequences, alignment, and phylogenetic tree structure in nucleic acid and protein sequences. MSA analyzes nucleic acid and protein sequences both structurally and functionally in a comparative manner. Nucleic acid and protein sequences are calculated with each other or with other sequences by means of MSA algorithms. In this way, it is possible to perform phylogenetic analysis, identify and regulate conserved regions, investigate homologies, infer protein structures, and observe mutations. Results and discussion. Sequence analysis of hydrolytic enzyme synthesis by rumen microorganisms, cobalt-3 structure and protein sequencing results of cellulose enzymes. For Fibrobacter succinogens, results on cobalt-3 structure and protein sequences of microorganisms belonging to Fibrobacter succinogens bacteria and given below. And this number IDs, strains were found to have more similar protein sequences than the CAA 61251.1 strain. There are 2 main groups and 1 outgroup with horizontal branches representing evolutionary lineage. Ruminococcus albus results on cobalt-3 structure and protein sequences of microorganisms belonging to R. albus bacteria are given below. There are 3 main groups present, as you see on the screen, and these numbers IDs, AAA, it starts AAA, and BAA, and also BAG, and BAG strains were found to have more similar protein sequences rather than other strains. Arflavifacins results on cobalt-3 structure and protein sequences of microorganisms belonging to Arflavifacins bacteria are given below. There are 5 main groups available, and it starts alphabet like AAB, CAB, CAC, and CAA, and SFX. Strains were found to have more similar protein sequences than other strains. Bactoclasticum results on cobalt-3 structure and protein sequences of microorganisms belonging to Bactoclasticum bacteria are given below. There are 2 main groups present. Ruminis results on cobalt-3 structure and protein sequences of microorganisms belonging to A. ruminis and bacteria are given below. There are 4 main groups present. E. oxidoreducens results on cobalt-3 structure and protein sequences of microorganisms belonging to this bacteria are given below. There are 2 main groups present, you can see on the screen. And Treponema saccharophyllum results on cobalt-3 structure and protein sequences of microorganisms belonging to Treponema saccharophyllum bacteria are given below. There are 3 main groups present as you on the screen. And S. ruminantium results on cobalt-3 structure and protein sequences of microorganisms belonging to this bacteria are given below. There are 3 main groups present as you see on the screen. These strains were found to have more similar protein sequences than the other strains. And bifermentans results on cobalt-3 structure and protein sequences of microorganisms belonging to this bacteria are given below. There are 3 main groups available, and these strains, it starts W and P, and these strains were found to have more similar protein sequences than other strains. And the last one is Prevotella ruminicola, results on cobalt-3 structure and protein sequences of microorganisms belonging to this bacteria are given below. There are many existing main groups. Here you can see the protein alignment and cobalt-3 structure. Phylogenetic tree of cellular sequences of all bacterial microorganisms, again, the cobalt structure and aligned protein sequences, results on cobalt-3 structure and protein sequences of microorganisms belonging to all bacteria are given below. There are many existing main groups. Since there was only one microorganism belonging to Clostridium longisporum, Vibrio fibrisolvens, and these all bacteria, cobalt-3 structure and protein sequencing finding couldn't be obtained. And you can see protein alignment on the screen. Cobalt-3 structure and protein sequencing results of xylanase enzymes. Bacterium cellulose results on cobalt-3 structure and protein sequences of microorganisms belonging to Bacterium cellulose bacteria are given below. There are 2 main groups present. The findings showed the strains. These 2 have NCBI IDs, have similar gene families and protein sequences. Ruminococcus albus results on cobalt-3 structure and protein sequences of microorganisms belonging to Ruminococcus albus bacteria are given below. There are 2 main groups present, and these AAT, BAB, and AAA strains were found to have more similar gene families and protein sequences than other strains. Ruminococcus flavifaciens findings on cobalt-3 structure and protein sequences of microorganisms belonging to Ruminococcus flavifaciens bacteria are given below. There are 2 main groups present, CAA, AB, CAA. Strains were found to have more similar gene families and protein sequences than other strains. Vibrio fibrisolvens findings on cobalt-3 structure and protein sequences of microorganisms belonging to Vibrio fibrisolvens bacteria are given below. There are 2 main groups present, AAD and also AAD 48 strains and AAD 483, and AAD strains were found to have more similar gene families and protein sequences than other strains. Prevotella ruminicola results on cobalt-3 structure and protein sequences of microorganisms belonging to Prevotella ruminicola bacteria are given below. There are many existing major groups. Under these results, it was observed that many strains had more similar gene families and protein sequences than other strains. And you can see the protein alignment shape on the screen. Treponema saccharophyllum results on cobalt-3 structure and protein sequences of microorganisms belonging to Treponema saccharophyllum bacteria are given below. There are 2 main groups available. It's observed that this group ID strains have more similar gene families and protein sequences. And some results on cobalt tree structure and protein sequences of microorganisms belonging for this longum bacteria are given below. There are two main groups present. It's observed that QNL and also QNL strains have more similar gene families and protein sequences. Since there was only one microorganism belonging to Pyrbacter succinogens and Lactobacillus acidophilus bacteria in this study, cobalt tree structure and protein sequencing findings couldn't be obtained. Conclusion, the need for bioinformatics tools is increasing by day by for various reasons, such as the ease of obtaining existing data by researchers uploading new data as new data are added, evaluating data in a biologically meaningful and accurate way, and discovering patterns in data. The fact that analyses that would take days can be performed in a few minutes with the help of bioinformatic tools has revealed the importance and necessity of in silico analysis. It was a part of my thesis analysis, and in this thesis, previously unexplored rumen microbial population derived hydrolytic enzyme genes, and the enzymes expressed by these genes were characterized using computer-based methods or bioinformatics tools. In the study, the data of hydrolytic enzyme genes in rumen microbial genomes were obtained, edited, and extracted from NCBI database, nucleotide and protein sequences of both cellulose and caselinase enzymes were determined and in silico analyses were performed. Phylogenetic tree structures, cobalt tree structure actually, were created by applying multiple sequence alignment method on nucleic acid and protein sequences on the data obtained, edited, and extracted with FASTA format from the NCBI database on the basis of cellulose and caselinase of the protein, and nucleic acid sequences of the bacteria investigated in this study. Thank you for listening. If you have any question, I can take kindly.
Professor, definitely you are having a lot of questions.
He knows everything about my thesis. Maybe you can ask something.
That's why he must be having so many questions from you.
He asked. He asked. 2 weeks ago, he asked me.
Okay, thank you.
You're welcome. Thank you for listening to me.
Okay. During this session, we have 5 to 6 presentations, and all the presentations are finished now. Now, the session is going to be end, and definitely that's all for today's program. Tomorrow there will be the program that will start at around about 9 o'clock. So hopefully meet you tomorrow at 9 p.m. a.m. Thank you.